Okay, so I think just we can just start for today. So others will join afterwards. Okay. Okay, so good evening everyone. So I think you can just see the slides and I am also audible. So welcome to the week 6 section of the NPTEL problem solving section of the course Cell Biology, Cell Law Organization, Division and Process. So my name is Isaac. I'm from School of Life Science, JNU, Delhi. I'm doing my PhD research at School of Life Science. So I think just we can move on to the first question. So does anyone have any doubt from the previous section that should be cleared out? Okay, so I think that's fine. So we will just start with the first question then. So coming on to the first question. So we will be discussing about 30 questions for today. So 10 from previous year and 10 from this year and 10 questions that I have made till now. So coming on to the first question. What role does cohesin play in DNA repair? It facilitates fidelity of homologous recombination by holding sister chromatids together. It repairs double-stranded breaks directly. It degrades damaged DNA or it prevents DNA replication error. Which might be that, sir? What role does cohesin play in DNA repair? One function we know, apart from DNA repair, there is also some other function that cohesin does play. I am getting option number 1. What role does cohesin play in DNA repair? Anyone else? I got option number 1. Okay, so if you look into option number 1, that's totally correct. But is it needed for DNA repair? Basically, it facilitates the fidelity of homologous recombination by holding the sister chromatic together. That's a different thing. But there is also another function that cohesin do play out. That is, it repairs double-stranded breaks directly in case of DNA repair. Cohesin do keep the together the sister chromatic together. So when they are together, chances are that the and when it is interacting with the other homologous chromosome, then it has the tendency to undergo homologous recombination. But that is not associated with DNA repair, right? It's a process called DNA, what is called as crossing over, that happens in meiosis. But when it comes to DNA repair, some of the other function that it does carry out, that it repairs double-stranded breaks directly, cohesin. So it acts as a motor protein at some point, and because of that particular function, because of the loop extrusion that it can carry out, the cohesin has the capability to carry out. It can also dictate double-stranded breaks, and it can also prepare those double-stranded breaks directly. So that's another function of cohesin other than just facilitating homologous recombination. So the other two functions are nullified in case of cohesin, so it does not carry out these two functions. It does not degrade damaged DNA, neither it prevents DNA replication error, but it do repair, that it do detect double-stranded breaks that is present within the DNA, and hence it has a capability to repair those double-stranded breaks directly also, apart from just facilitating the fidelity of homologous recombination. So I hope I am it is understood. So anyone has any doubt? If you have any doubt, you can type it out, or just you can unmute yourself and then ask. No issue. Okay, so I hope it's clear. So I will just move on to the next question. Okay, coming on to the next question. So the double-stranded DNA is wrapped around a histone octamer for how many turns? 1.46, 2.46, 1.67 or 1.77? Which might be the answer? DNA wraps around histone octamer to form a nucleosomes, right? So how many turns does a single DNA wrap around the histone octamer? It actually exists between 1 to 2 turns, but the correct value is between one of these options. Does anyone of you know? Even if it is wrong, it's okay. You can just try it out. Okay, so answer to this question is 1.67. So what happens is that in order to become compact, right, like in order to for the DNA to condense, there are certain proteins called as histones. The H1, H2, H2A, sorry, H2A, H2 can be H2A and H2B, H3 and H4. So this H2A, H2B, H3 and H4 actually join together form a histone octamer. So these are 4 subunits. So they exist as a dimer. So that makes up the 8 subunits. That means that there are 2 copies of H2A, 2 copies of H2B, 2 copies of H3 and 2 copies of H4 that joins together to form what is called as a histone octamer. So there are like 8 histones that contribute a histone octamer. That's why it's called as an octamer there. So in order for a DNA to condense or in order to become compact, what it does is that it wraps around at specific regions of the DNA interact with this histone chromosome, histone octamer, and wraps around it. So it's like a circular board. Consider the nucleosome like a spherical board in this case. So DNA starts to wind around its case. So when it winds around, it completes one winding, then it completes one turn. So then it starts to wind once more, not completing the whole 2-turn process, but in between 1 to 2 turn they actually bind with the histone octamer, and then there is a linker histone called as H1, which we will discuss afterwards. And through this interaction, the total winding that the DNA actually gone across the spherical nucleosome is about 1.67. So it completes one turn, then it winds a little more, and then it forms a 1.67 turn, not completing it up to a 2 turn. So that's why double-stranded DNA is wrapped around the histone octamer for about 1.67 times. 67 turns. So I hope this I am able to make you understand about this. So does anyone has any doubt regarding this? Okay, so I hope I'm clear. So I will just move on to the next question then. So coming on to the next question, which histone protein is not a core component of the nucleosome? H2A, H2B, H3 or H1? Which might be the answer?
Option D.
Option D I am getting. Any other option? Okay, I am getting option number 4 also. Yeah, I think majority of you agree with option number 4, right? So answer to this question is option number 4, that is H1 histone. It's actually a linker histone. It's kind of a linker protein, it's not component of the histone. So you can see the nucleosome structure here, right? So this is what you call as a histone octamer there. Leave the histone H1 there. This histone molecule consists of this H2A, H2B, H3 and H4, and these actually exist as two copies basically there. So that gives you a total of 8 core histone molecule. So then comes, in order to join, so the DNA actually winds there, right? So actually we have discussed that it winds around 1.6 times. So diagram as some issue, it looks like 2 tons and all. Okay, okay, no issue. So in order to keep the DNA attached to the whole nucleosome tightly, we need to like twine it into a nucleosome there, right? Twine it each other so that it won't leave that nucleosome core, this core histone octamer. So that function is carried out by this H1 histone there. So H1 histone helps combat chromatin by binding to a linker DNA between the nucleosome, bringing the entire and entering and existing linker DNA segments together. So you can see that if you consider this as the entering DNA segments, this is the link or exiting DNA segment, right? So you can see that they are joined together by the H1 histone there. So it keeps this DNA tightly wound to the histone octamer so that it does not fall off the histone octamer there. And this is primarily electrostatic in nature, with the H1 histones positively charged residues contributing to a balance of charge in the nucleus. So basically what means that this H1 histone, it is positively charged because of the presence of basic amino acid residues there. So that's what gives it the positive charge there. And because of this positively charged residues, the this is a nucleosome there. So negatively charged nucleosome actually interacts with each other, keeping them tightly together. And also by binding to the nucleosome core and linker DNA, H1 forms the chromatosome. So this whole structure is called as a chromatosome there, which aids in folding the chromatin into higher order structures like a 30 nanometer chromatin fiber. The whole length of the chromatin fiber that is made is approximately about 30 nanometers. So not only this particular portion, several other portions of chromatosome is present there that in turn compacts the whole DNA into what you called as this particular 30 nanometer chromatin fiber. It controls the accessibility of the linker DNA to DNA binding protein, influencing chromatin folding and gene expression. Transcriptionally active regions often have lower H1 compared to inactive region, because whenever this nucleosome is present there, whenever the DNA is slightly wound to a histone octamer, what happens is that the region becomes transcriptionally inactive. So that particular region is also called as heterochromatin, right? So whenever this particular region or that particular DNA or gene present at this particular region needs to be active, it needs to unwound itself. So H1 histone should not be there. If H1 histone is there, then it will become inactive. It is tightly bound to a nucleosome, right? So other transcription factors or any other protein cannot come to there and bind to it. So that's an issue there. So H1 histone should not be there at that time. So it also contributes to DNA stability and chromatin compaction, which can affect the DNA repair process. Upon DNA, these are some additional information that I am just giving. Upon DNA damage, H1 can undergo modification and release of chromatin. Under modification like acetylation, phosphorylation, citrulation that we will be discussing is also there. That might affect the interaction of H1 histone with the nucleosome there. So yeah, so I hope it is understandable. So anyone has a readout? Okay, so I think it's clear. So I will just move on to the next question. So coming on to the next question, which of the following correctly describes heterochromatin? Transcriptionally active and loosely packed, transcriptionally silent and tightly packed, contains only euchromatin region, found exclusively in prokaryotic cells. Which may be the answer?
Option 2.
Yes, I am getting option number 2. Okay, option 2. Any other option? Yeah, so basically I think each of you goes to option number 2, right? It's basic answer. So you are basically right, transcriptionally silent and tightly packed. So basically this is the differentiation between a heterochromatin and euchromatin. You can see that the heterochromatin is actually, these are the nucleosome, the DNA you can wound around the histone octamer, and similar in this case also. But the difference is that in one case, in the heterochromatin, you can see that it is in a tightly packed stage. You can see that there are several deacetylated histone tail, methylated CpGLNs, etc. Several other proteins that are there but are not binding to the transcriptional start site and all. But in case of euchromatin, you can see that it is in a more open conformation, right? So you can see that several of the methylation and also acetylation are present there. Demethylated regions are present there, and acetylated histone tails are also present there. So when it is acetylated, you can see that the histone tails are actually popping out, meaning the interaction is getting affected there. And also the transcriptional factors RNA polymerase 2 actually for needed for transcription and all are actually binding to the transcriptional start site because it is free, and also other proteins that actually prevent the binding of this transcriptional factor site are usually absent there. So that's why it's binding to it and starting the transcription. So whenever this close conformation is present there in case of heterochromatin, it means that this particular site where this heterochromatin is present is transcriptionally inactive and it is tightly packed there. While in case of euchromatin, whenever it is open, in an open conformation, it means that like the proteins can come to it and bind to it. If it is a transcription factor, it can initiate the transcription and all, and hence can undergo the mRNA generation from this particular region of DNA where it is binding. So that's the basic difference between heterochromatin and euchromatin. So if you are staining this particular region, we can see that the heterochromatin actually appears as a dark band because of the presence of tightly packed region there. So it is stained densely there. While in case of euchromatin, if you compare it with heterochromatin, it will be lightly stained because of the absence of density that is present there, because it is it has a loose conformation there, right? So stains won't be densely packed there, so it will appear as light bands there. So the difference between the dense bands as well as the light bands give you an idea about at which site of the DNA is actually undergoing transcription and all. So that's the region. Contains only euchromatin region. That's not a correct answer. Euchromatin means like it is transcriptionally active, right? And first exclusively in prokaryote. That's not right. So the answer to this question is heterochromatin are transcriptionally silent and consist of tightly packed DNA region there. So does anyone have any doubt? Okay, so I think it's clear. So it's a basic question, right? So I will just move on to the next question. So coming on to the next question, which histone variant is specifically associated with centromere? H2AX, H3.3, SENPA, or H1? Which might be the answer? Okay, I'm getting option number 3. Any other option? Okay, so basically you are right. So the answer to this question is SENPA. So basically there are several histone variants because you know that there is H2, H3, H1, H4 like that, right? So basically these act as different histone variants, and these variants are found in different places of chromosomes within a same organism or in different organisms. The histone variants might be present specifically associated within that organism itself. So if you look into this particular variant, that is SENPA, it's actually a variant of H3, histone H3. So it is one of the highly distributed variant of histone. So and these are found associated with centromeres. So it is crucial for the formation and function of centromeres, serving as foundation of kinetochore assembly and ensuring proper chromosome segregation during mitosis. So basically kinetochore in centromere, the kinetochore is actually present there, right? So that mitotic spindle or any other meiotic spindle and everything attaches to it, and then it helps in cystochromatic separation, right? So So this SENPA is actually a variant of histone H3 that is present at this particular region, which helps in the proper chromosomal segregation and all. So it is incorporated into a centromere chromatin, marking the site where kinetochore is assembled. So there should be something that initially that should bind to a centromere that where the kinetochore should be attached to the right. So this particular variant of H3, that is SENPA, actually helps to promote this particular function, and it is essential for assembly of both inner and outer kinetochore components. You know that the kinetochore actually exists as not as a single fraction, but it consists of two components, inner as well as outer kinetochore components. So as far as I recall, the mitotic spindle actually attached to the inner kinetochore components. Please check that out also. And also, SENPA containing chromatin is recognized by proteins. Other SENP proteins are also present there, including SENPN, which recruits other constitutive centromere associated network components like SENPC and SENPI. So these are some additional information that is given to you. Basically, if you can note it down, you can note it down, but it is not that important. But it always good to know like some of the additional factors in this case. So these proteins are crucial for stabilizing SENPA and recruiting additional kinetochore components. This constitutive centromere associated network, which is recruited to the SENPA nucleosome, serve as structural core assembly of other kinetochore components like this KNL1, MIS12, NTC, AT. So these are components of kinetochore there. So once these components are attached, the kinetochore components start to assemble near the centromere, forming what is called as a inner and as well as outer kinetochore. So that's the basic function of SENPA, which is a variant of H3 syndrome histone there, and it is specifically associated with centromere, and which is basically helps in kinetochore assembly, which is usually involved in chromosome segregation. So if we look into the other two components, that is H2AX is involved in DNA repair and gene of stability, and H3.3 is associated with active genes and region of high histone turnover. High histone turnover means that there are certain regions that are present within the nucleosomal DNA, which is transcriptionally active at a higher rate. So some point of time it needs to be compacted, but at majority of the time it needs to be free. So the histone turnover, that is the synthesis of histone, should be less. The presence of histone at that region should be less as compared to the degradation of histone that is present there. So histone that region must be free from histone at that time. That's why it's called as region of higher turnover and is associated with region of active genes. So whenever active gene is present there, in order to make it active, the histone should be absent at a particular point of time at majority of the time there. So that's why it's called as H3.3 is associated with the regions having high turnover now, histone turnover. So yeah, so this is about the SENPA like H3 variant of histone variant of H3 histone there. So I hope I am clear. So anyone has any doubt? Okay, so I think it is quite clear. So I will just move on to the next question then. Coming on to the next question. The DNA that wraps around the nucleosome or the histone octamer is how many bases long? 155 bases, 147 bases, 165 or 167 bases of nucleotide, which may be the answer. So you know that the histone actually, okay, option number 4 I am getting. We know that the histone actually binds around the 1.67 turns are there, right? So what might be the length of the bases that would be wrapped around each histone octamer? Okay, option number 4 I am getting. Okay, so the approximate answer to this question is 147 nucleotides or bases. So not 167, it's 1.67 turns that is there, but in order to compare that, there is actually the approximate value is about 147 bases or 147 nucleotide. So yeah, so I think the answer is clear. Does anyone have any doubt in this case? Okay, so I think it's clear. So do check it out also. Some of the mathematical equations there. So I will just move on to the next question then. So coming on to the next question, which portion of the histone undergo majoritarily the amino acid modification? The C-terminal tail, N-terminal tail, both the C-terminal as well as N-terminal tail, or none of the above? Which might be the answer? Yeah, I'm getting option number 2. Any other answer? There are several modifications that are happening within a histone tail, right? So amino acid modification to take place. Some of them are acetylation, phosphorylation, etc. So at majority of all the histones, if you look into it, at a certain portion of the histones, this modification take place. So it can lead to either activation or repression of certain activities, including transcription, etc. So at which end of the histones does this amino acid modification take place? Okay, so I got answer number 2. So basic option number 2. So that's the that's the answer. So N-terminal tail of the particular histone, whether it might be H4, H3, H2A, H2B, which are components of the core nucleosome complex or histone octamer, which forms the histone octamer there, actually undergoes this N-terminal modification there or amino acid modification there. So you can see that the tail region of the C-terminal end is actually found inside which interacts with each other, while the N-terminal, so that the histone octamers can be interacting with each other, while the N-terminal is actually bound to the outside. You can see that the N-terminal of this particular H3 histone is much longer as compared to the outside. And also apart from that, thus if you look into a histone H2A and H2B, you can see that do modification also take place in case of ubiquitination. Ubiquitination is used for protein degradation, right? So whenever this particular amino acid this lysine that is present there is ubiquitinated, this particular histone H2A as well as H2B can undergo proteasomal degradation there. So that's why ubiquitination happens in case of C-terminal end of these both heptamers, that is H2A and H2B. Some amino modification do take place in case of acylation take place in lysine. However, if you look into majority of the amino acid that is modification that is taking place there, you can find that all the N-terminal end of this particular histone octamer undergoes this kind of amino acid modification there. You can see phosphorylation there, methylation there, citrullination, acetylation, at specific amino acid ratios. Serine undergoes phosphorylation, including this what you said, threonine there, right? So lysine undergoes methylation also, acetylation also. Lysine primarily undergoes acetylation and all. Histidine is also present there that undergoes phosphorylation. Citrullination is undergone by this, I think, arginine there. So basically, these are modification that take place at the N-terminal end of the histone octamer. But in case of H2 and H2B, the C-terminal is also free in this case. You can see that it can the C-terminal majorly undergoes ubiquitination, but at some point in case of histone H2B, from what we know till now, acetylation of lysine can happen there, which can also influence the activation and repression of certain genes and everything. So if you see majority of the commonly in case the N-terminal tail, this is called as the tail region of the histone octamer or individual histone there, that undergoes amino acid modification including acetylation, methylation, citrullination or phosphorylation. And some point of time ubiquitination can also happen, which indicate that the histone protein needs to undergo proteasomal degradation. So that's the basic answer. So does anyone have any doubt? Okay, so I think I'm clear. So I will just move on to the next question then. Okay, coming on to the next question. Okay, this basic question. Which technique is used for real-time imaging of DNA loop extrusion by SMC complex or structural maintenance of chromosome complexes? X-ray crystallography, atomic force microscopy, single molecule fluorescence microscopy, or Western blotting? Okay, I'm getting option number 3. Any other option? Yeah, I think majority of you go with option number 3, right? Single molecule fluorescence microscopy. So this we will discuss also later also. So you can see that this is condensing and cohesing. You can see the structure is almost the similar in this case, right? So there is a hinge region that is present there. This is the ATPase hydrolyzing region that is present there. Other proteins are also bind to it, and it exists as a each of the coil region exists as a coil dimer there, right? So this is from individual protein molecules. This is also from individual protein molecule that coils to in a like independent manner to form the particular structure. And you can see that SSC3 is also present there. Sorry, SSC1 is also present there, which binds the two dimers together so that it can cooperate with the chromosomes and all. Okay, so you can see that this is a image that is obtained from single molecule fluorescent microscopy. So you can see that the different time in seconds is given there, and yeah, so you can see the slightly extension there, right? So that what happening in a diagrammatic view, it is given in the bottom lane. So you can see that whenever the condensing or coercion is present there in a particular region, you can see that the sharp glow that is present there, right? Meaning that the DNA is getting concentrated there. So the condensing or coercion is actually coming into this region, binding to it, and what it does is that it pulls the DNA to the outside. When it binds to this particular region, you can see the coercion binding there or condensing binding there, then it starts to extrude out the chromosome to the outside, forming a loop. You can see that whenever the loop is getting increased there, these are DNA loop there. So the coercion is binding there because of the motor protein activity. The coercion and condensing actually hydrolyzes ATP, and it has a motor protein like activity. It can change the structure of the DNA there, so making loops and all. So you can see that the coercion is binding there, and you can see that the DNA is getting extruded out or pulled out. You can see that it gets pulled out and forming a particular linear loop there, and it which each time point up to 80 seconds you can see that the loop length is getting formed there. So this is the diagrammatic view what's happening. There is a free DNA there, the condensing or coercion starts to bind to it, and it pulls out the DNA from either one end or from both ends to form a loop by utilizing ATP there. So if you look into a condensing structure, it can carry out one-sided loop extrusion. In case of coercing, it is also a motor protein, not just attacking the sister chromatids together or having a DNA repair like activity. It can undergo two-sided loop extrusion. It pulls the DNA from both the sides so that the loop can form at this particular region, and with time the loop size gradually gets increased. So this particular real-time image is done by this particular single molecule fluorescence microscopy. Any other fluorophore can be tagged to it, so that's why it gets colored there, and because of the high intensity of DNA that is present there, it is more colored towards the red there. So okay, so can any one of you tell me what is this X-ray crystallography, atomic force microscopy, and Western blotting does? Any one application or a single definition, if any one of you know? Why is this involved, and what are these uses, application, and everything? Okay, so yeah, I will just give you explanation there. So X-ray crystallography means that you have to determine a three-dimensional structure of a particular protein there, right? So if you have purified protein, and suppose that you have purified a protein from a bacterial expression system, and then you have made a crystal of that particular protein there. So if you have a crystal, then you can undergo a process called as X-ray crystallography. So you put a crystal in that particular instrument there, and then pass an X-ray beam through it. So X-ray is actually at the wavelength of electrons there, right? So what it does is that it whenever hits a particular region of the protein molecule in the crystal, it starts to diffract out. And based on the diffraction angle and the diffraction intensity that you are getting, you can actually map out the three-dimensional structure of that particular protein that is present in the crystal there. So it is used to determine the three-dimensional structure of that particular protein. Atomic force microscopy or AFM force microscopy, meaning that if you want to determine the overall morphological characteristic of a particular surface or any material, you can actually use atomic force microscopy. So I think it's better to check within the Google and all to find out how the proper mechanism of atomic force microscopy is. And Western blotting is another method that is used to determine the presence of a particular protein within a protein sample. So it is used for protein determination, and also you can use several antibodies that can be targeting a particular tag present in the protein, or the antibody that is interacting with that particular protein itself, and then do a Western blotting analysis to determine whether in that particular sample that particular protein is there or not. And yeah, so that's the basic functionality of this particular technique. So anyone has any doubt regarding this? Okay, so I think it is quite understandable. So I will just move on to the next question then. Coming on to the next question. Which process powers the loop extrusion activity of SMC complexes? ATP hydrolysis, GTP hydrolysis, NAD plus hydrolysis, or formation of a proton gradient? This may be the answer. Okay, I'm getting option number one. Option number one. Yeah, basically you are right. So the answer to this question is ATP hydrolysis. So basically this is a structure we have discussed before, so you can just see it for your reference. So this particular region, that is this region that is bound to different proteins, right? So it has the capability to hydrolyze region. So this is called as head region, and this is called as a hinge region. So it can actually hydrolyze ATP. So the process that powers the loop extrusion activity is by ATP hydrolysis. The SMC complex, including condensing coercin and other proteins like SMC5 or 6, utilize the energy from ATP hydrolysis to extrude DNA loops, which is crucial for DNA organization and regulation, as well as DNA combustion or condensation. While ATP binding itself is involved in initiating the twist during loop extrusion, it is the hydrolysis that drive the actual extrusion process. It initiates the process DNA binding. However, in order to make the loop extrude out, DNA hydrolysis need to happen. So one is the DNA ATP binding, and another is the ATP hydrolysis. ATP hydrolysis actually powers the loop extrusion and all. So that you need to remember in mind. So ATP hydrolysis that is happening within the head region of this this SMC complex, that is structural maintenance of chromosome complexes, actually powers the loop extrusion or formation of loop region because of the motor action that is taking place within the condensing and coercin molecule. Okay, so that is, I think, quite understandable. So can any one of you tell me where this formation of proton gradient happens? Like what does this lead to? Formation of a proton gradient. Any other process that might not be involved in this SMC complex and all? Where does this happen and what does it lead to? Proton gradient formation. Okay, so I think you have learned about ATP generation and everything, right? ATP synthesis actually it takes place in mitochondria with the help of a protein called as ATP synthase, right? So formation of ATP, this proton gradient within the inner mitochondrial membrane and or intermembrane space actually drives out the actually make the ATP synthase functional, and from that it helps to form the ATP from the like what is it the because of the flow of like proton gradient, that is proton high concentration from its high concentration to the lower concentration. So that acts as a motor protein that helps in the formation of ATP. So in this case, in that case, ATP generation is taking place, not ATP hydrolysis. So in this case, in order to power the loop exclusion activity, ATP hydrolysis needs to take out. So I hope I am clear. Does anyone has any doubt? Okay, so I hope I am clear. So I will just move on to the next question then. Coming on to the next question. What is the length of a combatant nucleosomal DNA? 30 nanometer, 40, 35 or 37 nanometer? This we have discussed earlier. Which might be the answer? Yeah, I'm getting option number 1. Yeah, basically you're right. I think we have discussed this before, right? So the answer to this question is 30 nanometer chromatin fiber. So it condenses around like each DNA contains around 1.67 times, and throughout each turn there will be approximately about 147 nucleotides that will be present within each histone, that is wound around each histone octamer. And then this happens multiple times, and so the length of the chromosome is actually getting condensed into a structure called as genomic DNA is actually getting condensed into a structure called as a chromosome, right? So this chromosome fiber actually is of length. If you look into a total fiber length of this chromosome, it is condenses up to 30 nanometer. So the three-dimensional structure, so that three-dimensionally fits into a particular structure called as chromosome. So that's the basic answer there. So does anyone have any doubt regarding this? So, or should I explain it once more? Okay, so I think I'm clear. So I will just move on to the next question then. So coming on to the next question. What may happen when SMC protein or structural maintenance chromosome protein fails to function properly in cells? Chromosome contents more efficiently, chromosome missegregation and genome instability happens, the DNA replication speeds up, or cell enter apoptosis immediately. Which may be the answer? Okay, I'm getting option number 2. Any other option? Yeah, I think each of you agree with option number 2, right? So it leads to chromosome segregation and genome instability. So SMC protein or structural maintenance of chromosome proteins play a crucial role in cystochromatic cohesion and chromosome condensation. The examples include what we have discussed, like condensing, cohesin, SMC5 or 6, right? So these are proteins that are SMC, also called as SMC protein, and they are involved in cystochromatic cohesion and chromosomal condensation. So if they fail, what happens is that if some mutation happens to this protein, or if there is some deleterious effects that is happening in the translation or transcription of proteins, etc., it can lead to missegregation and all. And also this results in genomic instability, which is characterized by changes in the number of structure of chromosome, potentially leading to conditions like aneuploidy. So basically what happens is that without this particular combustion into that particular 30 nanometer chromatin fiber or the chromosomal arrangement, what happens is that the segregation of chromosomes won't happen properly, because the centromere should be present there and each and every telomere should be there, and the other regions, the chromatid arms should be properly maintained there. So only then the histone variants like SENPA comes to there, then it recruits kinetochore, the inner and outer kinetochores there, and afterwards only kinetochore then comes into place, its own specific position there, then it can lead to other processes like mitosis and mitosis, including the metaphase stage as well as anaphase stage, right? So without the proper functioning of these SMC proteins, the loop exclusion cannot take place. Cohesion, these processes cannot take place also. So as compared to that, that's one function. It can lead to this particular condition like aneuploidy. Other than that, as we have discussed before, it is involved in DNA replication and a recombination process, because cohesion involved in increasing the fidelity of the homologous recombination between sister chromatids there, right? And also it also helps in DNA repair, the first question that we have discussed here. And this can also lead to more DNA damage as well as genome instability and everything. So this SMC 5 or 6 that you have discussed, rather than the condensing and coercing molecule, motor proteins, helps in maintaining the replication force stability. And it can, if some failure happens to this, it can increase the replication stress. It can also reduce the replication that is taking place, and also it can also causes the chromosomal segregation and everything. And even though if you look into the, so in this case you can see that option number 3 is wrong, because it can actually reduce the speed of the DNA replication because of that, and also it cannot condense properly because of the failure of this SMC proteins and everything. And apoptosis does not happen immediately. However, apoptosis can happen if it is identifying that there is a problem there, but it takes some time for the cells to recognize that okay, this cell is going some problem, so it needs to undergo cell death or apoptosis. So it's not like it immediately undergoes apoptosis. It takes some time. It has to go through some checkpoints and everything, and then afterwards only it undergoes apoptosis and everything. So that's one thing in this case. So I hope this answer is clear. So the answer to this question is, if SMC proteins fail, then it can lead to chromosome segregation and genome stability. That's the most appropriate option out of these 4. So anyone has any doubt? Okay, so I think I am clear. So I will just move on to the next question then. So coming on to the next question, what role does the protein WAPL or WAPL play in cohesion dynamics? Loading cohesion into a DNA, stabilizing cohesion on the chromatin, unloading cohesion from DNA, or cleaving cohesion during anaphase. Which might be the answer. What does the role of WAPL is like? What does WAPL do in case of cohesion dynamics? Okay, so I think I will just give you the answer in this case. So answer to your question is that option number 3, unloading cohesion from the DNA. So this is the functionality of WAPL there. You can see that in case of this is the cohesion or condensing, whatever it might be, this is the cohesion molecule. So you can see that the SEC1 is actually present there, and each and everything is present there. So this is in a like a closed conformation there, right? So this is bound to the DNA. So this WAPL protein along with this PDS5 together with the help of ATP hydrolysis binds to this particular region there, and then separates or cleaves out this particular SEC1 protein that is binding together these two proteins together. So basically it gives a close conformation there, and when ATP hydrolysis take place there, what happens is that this particular structure gets opened up. So it is actually having the first it actually interacts with this particular cohesion molecule, and it brings about this particular what is a close conformation or tightly closed conformation. Afterwards, when ATP hydrolysis happens, this cohesion gets released from the particular DNA molecule, because you can see that the SEC1 molecule is getting open there, right? So whenever it is open, this cousin is in an open conformation there, so it is free to leave from the DNA. So that what it does, it unloads the cousin from the DNA molecule. So a WAPL along with this particular protein PDS5 and ATP hydrolysis actually does this process, the unloading of cousin from the DNA. So it acts as a cousin release factor by interacting with the cousin complex and promoting its dissociation from the chromatin. This process is essential for the proper resolution and separation of cystochromatic during the mitosis, because it opens up the whole cousin molecule there. It achieves by opening the DNA exit gate in the cousin rings, this particular region there from the ATP or head region there, allowing the cousin to release from the chromatin. So that's the particular function of this particular protein, that is WAPL or WAPL proteins there. So does anyone have any doubt regarding this? Okay, so I think I'm clear. So I will just move on to the next question then. Okay, so coming on to the next question. Majority of amino acid modification take place in which histone moiety? H4, H2A, H2B or H3? We have discussed this before, which may be the answer. Majority of amino acid modification including acetylation, phosphorylation, etc. take place in which histone moiety? Okay, so I suppose I will just give you the answer. So answer to this question is H3. So the modification that we have discussed before take place in the N-terminal tail. At some points it also take place in the C-terminal tail of H2A and H2B, right? Majority in the H2B histone molecule there. So you can see that the length of the H3 N-terminal tail is much more higher as compared to the other N-terminal lengths there, right? So it is shortest for the H2A in this case, and amino acid modification do change according to that also. And also if you look, all the modification that we know happens within histone molecule, including phosphorylation, methylation, acetylation and citrullation, take place in this particular N-terminal tail also. And also if you look into the length of the tail, it is comparatively much higher as compared to the other N-terminal tail of other histone molecules there. So if you look into a particular structure, you can diagram, you can see that the histone modification is higher in case of the N-terminal array chain of the histone H3 molecule there. So I hope I am clear. So does anyone have any doubt in this case? Okay, so I think it is quite clear. So I will just move on to the next question then. Okay, coming on to the next question. Which protein is known to work in conjugation with cousin to define the boundary of topologically associated domain, or TAD? Condensing, WAPL, NIBPL or CTCF? Which might be the answer? Which protein is known to work in conjugation with cousin to define the boundary of topologically associated domain, or TAD? Okay, so I think I will just give you the answer. So the answer to this question is CTCF. So it is a protein that forms homodimers and bind to specific DNA sequence at the TAD boundaries, where it interacts with the coercin complex to block loop extrusion and thus define this boundary. So we have discussed about loop extrusion, right? So it is carried out by this SMC protein. In this case, condensing is that SMC protein, and it uses ATP hydrolysis to form loops, right? And also we have discussed about WAPL. So WAPL is a protein that allows it act as a coercin release factor there, right? So it helps to release the coercin from the loop region there. So if you look into a normal functioning of a loop extrusion, this is actually how it happens. So suppose that in this case the DNA is free and it is then interacting with this condensing molecule there, which is a motor protein. Because of the motor protein activity there, you can see that a loop forming there along the particular region of the DNA there. So it actually then goes on until it recognizes certain region called as TADs. This is the CTCF binding site or TAD region, topologically associated domains there. So it is bound by this particular protein CTCF. So TAD regions are present there, and it is bound by this particular region CTCF proteins there. And whenever this condensing or coercin or SNC protein detect this, the loop extrusion stops there. It won't go beyond that particular region. But whenever coercin is happening, coercin is present there. In this case, you can see that the loop extrusion is not from there because motor proteins are present there, right? And whenever this no CTCF is present there, CTCF means that it is a protein that recognizes TAD region there, right? So the TAD region is present there, but there is no CTCF binding to it. So what happens is that the loop extrusion continues out. Even the TAP region, this particular TAD region is going into it. Into this particular site, you can see that the loop extrusion is getting continued because of the absence of the CTCF there, right? So the loop formed will be much more in length as compared to what happening here in this case, because the TAD region is also going in and it's continuously there. And also WAPL is also not there. When WAPL is not there, what happens is that the coercin actually don't know when to release there, because WAPL is actually a coercin release factor there, right? So whenever the WAPL is absent there, it not even recognize the CTCF protein there, not or that is found in the TAD region. It continuously extrudes out the loop there, because the coercin is not getting released there. It undergoes ATP hydrolysis and the loop extrusion continues on there in this case. And PDS5A is also a accompanying factor of WAPL there. So we have discussed that before. It is also similarly related to what you called as a this release of coercin from the molecules. So similar results is there, even though the CTCF factor is the CTCF protein is bound there, the loop continues on extruding out. So that's the function. So when no CTCF is there, the loop extrudes out without recognizing the TAD topologically associated domains there. And yeah, so that's the function of CTCF. It actually recognizes the region there, so that it tells the coercin that you need to stop the loop extrusion. It has reached the topologically associated domain or TAD region there. So that's the function of CTCF there. So anyone has any doubt in this case, or should I explain it once more? Okay, so I hope I'm clear. So I will just move on to the next question. Okay, coming on to the next question. SMC proteins are known to form which type of structural organization? Tetrameric globular complexes, dimeric coil structure, single stranded helices, or beta sheet barrels? Which may be the answer? You have seen the structure of coercin or condensing, right? So there's a kind of a structure that defines its boundaries. The protein actually consists of a specific three-dimensional structure there in both the cases. What structure might that be? Could you please repeat? You can just type it out if you have any network issue, Andrew. I'm getting option number 2. Any other option? Yeah, I think everyone goes with the dimer coil structure here, right? So yeah, basically it is known to form dimer coil structure. So you are basically right. So basically, if you look into an individual structure, I could have shown one another structure diagram also there. Okay, anyways, no issue. So this is the hinge region there. So these are two different proteins. One is the SMC3 and other is the SMC1. You can define by the differentiate by the colors there. So this is the hinge region. So one coil region is present protruding out like this, and one coil region is protruding out this. Sorry, one linear region is protruding out this. So one to the one end, other to the one end, and hinge region actually stays here. And the property of this is that the two regions that is pointed out to both the ends has the tendency to coil around each other and to form this three-dimensional coiled end structures there. So the end of each region consists of this particular head region there, and they join together to form a dimeric coil structure. So you can see that this is a coil structure and it is dimeric in nature. If we consider of three regions there, then it will be trimeric coil structure. So that's the functionality of each individual SMC protein there. So you can see that in this region that also happened. The hinge region consists of two regions that are protruding out with the head region at the bottom. Then the coil on each coil along with each other to form this dimeric coil structure there. And the hinge region of the both the SMC proteins actually interact with each other and then form this hair clip like structure. And this hair clip like structure is interacted with each other by this SCC1 protein or RAD21 protein along with the presence of other other proteins that bind to it, the WAPL, PDS5 that you have discussed before. These are all release factors there, right? So this is actually the structure of the structural configuration of this SMC protein. That if you look into one of the SMC protein, it is dimeric, it has a dimeric coil structure there. So does anyone have any doubt? And it is also antiparallel in nature. So to check out what is antiparallel in nature, you will get the answer. So anyone has any doubt? Okay, so I hope I'm clear. So I will just move on to the next question. Coming on to the next question. How many histone variants of H2A is seen in case of sea urchin? 4, 5, 3 or 6? Which might be the answer? This answer you might have seen in the video, which might be the answer. How many histone variants of H2A is seen in sea urchin? Okay, I am getting option number 2. Any other option? Okay, so basically you are right. The answer to this question is 5. So basically we have discussed several other histone variants, right? One of the histone variants of H3 is SENPA, which is associated with syndromia, and it helps in kinetochore assembly and everything, right? Which helps in DNA chromosome segregation and all. So H2A, other histone variant histones also have some histone variants. However, some exceptions are there. Some of the histone do not show any variants and all. So that will go with afterwards and all. So in case of sea urchin, there are about 5 histone variants of H2A that are seen there. So I hope like it is understandable. It's basically something that you should memorize. So I will just move on to the next question then. Coming on to the next question. Okay. So in the beads of string structure, the beads are, what does the beads represent? The nucleosome core particle, SMC protein complexes, DNA polymerases, or none of the above? This is a basic question. You know the answer. Okay, so the answer to this question is nucleosome core particle. So I'm just giving a brief description on this, like you know the answer basically, but I'm just giving you an answer to it. So the beads on string structure in chromosome forms through organization of nucleosome, which are functional unit of chromatin. You can see the histone octamer there in each case, and the DNA is wrapped around it. And the histone octamer consists of these two copies of each of H2A, H2B, H3, and H4 to form a nucleosome core particle there. And this core particle is approximately 10 nanometer in diameter and consists of about 147 base pair that is bound to it each other there, right? So it wraps around like 1.67 times, and each wrap consists of about 147 nucleotides or base pairs of the DNA that are wrapped around each other. And each nucleosome is connected by a segment of linker DNA, this particular region you can see there, and which is about 20 to 60 base pair long. And this linker DNA acts as a string. You can see that it acts as string there, right? And this acts as the bead there. And so the whole model is called as a beads on a string model there. And when chromatin is in least compact form, it is called as a euchromatin. The nucleosome linker DNA gives rise to a capsid based on string structure there under electron microscopy, and it is this particular structure is dynamic and contains further into a more compact form during cell division to form what is called as a chromosome there. As the chromatin iones, the beads on string structure can coil into the 30 nanometer fiber that we have discussed before, which is a more compact form of chromatin organization. Okay, so you know the basic description there, right? What I just told. So yeah, so the basic answer to this question, in the beads on structures, the beads are called as nucleosome core particle. So does anyone have any doubt regarding this? So okay, I think I'm clear. So I will just move on to the next question then. Coming on to the next question. The nucleosome core particle contains a histone. Which one of the answer? Octamer, heptamer, nonamer, or decamer? It's also a basic question. Anyone? Yeah. So I'm not discussing that deep into it. You know the answer. It is a histone octamer. So okay. This we have discussed before. So this is a nuclear particle. You can see that there are 8 histone molecules, 2 copies of each, H2A, H2B, H3, and H4. And where H1 acts as the linker histone molecule there, right? Okay, so I think this is clear. So I will just move on to the next question. Coming on to the next question. Which histone protein does not show any kind of variance? So H4, H2A, H2B, or H3? There are several variants of histones there, right? So some of the histone does not show any kind of variation there. Out of this option, which may be the answer? Okay, I'm getting option number 1. Any other option? Anyone has anything else in mind? Okay, so you are basically right. The answer to this question is H4. So it is one of the slowest evolving protein. Like it is not it is highly conserved in nature because it is slowly evolving, right? So it is highly conserved in nature. It's no functional variance in the majority of the species. Although in case of trypanosoma, one variant like H4.V is seen in this case. So this is one exception there. However, in most of the cases, this histone H4 is actually highly conserved in nature and is slow evolving in nature. That's why it doesn't have any functional variant in nature. If you compare the other histone, H2A has the highest number of variants, including all these names that is given there. That is H2A dot X, H2A dot Z, macro H2A, and other variants are also seen in this case. So among these, H4 has the least number of variants, while H2A has the highest number of variants that is known till now. And also H2B also has a few variants that is come in between the H2A as well as H4, but it exhibits still some diversification in this case. And also H3, as we have discussed before, some of the variants include SENPA, which is sorry. Okay, so sorry for that, there was some network issue. So H3 actually exists as different variants like H3.3 or SEN H3, which is also called as SEN PA in case of humans, and it shows higher degree of variability in this case. So SEN P we have discussed before, it is binds to centromere, and once it binds, it initiates the kinetochore assembly. It binds to other SEN P proteins and everything, and it also initiates the kinetochore assembly, which is involved in chromosome segregation and all. So that we have discussed earlier before. So in this case, the histone protein that does not show any kind of variant is H4 histone, and H2A in this case shows the higher number of variants among the histone proteins and everything. So I hope I am clear. So anyone has any doubt in this case? Okay, so I think I am clear. So I will just move on to the next question. So SIR2 is a HAT, HDAC, or PI3K, or none of the above. So which may be the answer? SIR2 is a protein which can be a histone acetylase, histone deacetylase, a kinase, or none of the above. What type of a protein is it? SIR2. Okay, so the answer to this question is histone deacetylase. HDAC stands for histone deacetylase, and HAT stands for histone acetylase. So basically, it consists actually is protein, and it consists of its own mammalian homologs known as sirtuins, which ranges from SIRT1 to 7, and these uses NAD plus dependent deacetylases. So in case of coercin, what happens is that it uses ATP for loop exclusion and everything, right? Coercin or condensing, whatever it is. So it uses ATP hydrolysis to carry out the function. But this particular family of sirtuins, that is SIR2 belongs to the sirtuin family. It is NAD plus dependent deacetylases that removes the acetyl group from histones and other proteins, playing crucial role in transcriptional regulation and several processes, or including cellular processes such as genome stability and lifespan extension. In this case, genomic lifespan extension, degradation should be prevented there, right? So these have this kind of like activity and everything. So from acetyl groups are usually present within the lysine molecule, amino acid there, right? So it removes this kind of deacetylate, that is SIR2 removes the lysine molecule from this particular, the acetyl group from this particular lysine molecule. It comes under this class 3 deacetylase. There are different type of class. I think there are, to what I recall, there are 5 classes of histone deacetylases. Please do check it out if I am right or wrong. So distinguishing them from other classes due to their requirement of NADH cofactor. So apart from this particular classes, and this class is distinguished separately from the other classes because it uses this NAD plus as a cofactor. You can see that it is because of the fact that it's a NAD plus dependent deacetylase. And by maintaining the silencing of telomeres and the region, SIR2 helps in preventing inappropriate gene expression and recombination events that could destabilize the genome there. So the deacetylation actually prevents the gene expression, and it can acetylation can actually at most cases it can either prevent or promote gene expression there, right? So in this case deacetylase that like SIR2 actually prevents the gene expression that is happening unwantedly there, and also it prevents the unwanted recombination events that are taking place that destabilize the genome. And majority it is present near the telomeres. It silences the telomere region to inhibit these two activities, and also in other repetitive regions also. So that's the basic answer. So SIR2 is actually a NAD plus dependent deacetylase that belongs to the sirtuin family, and it is characterized because it uses this NAD plus as a cofactor there, and it is primarily found at telomeres and also in other repetitive regions. So what it does is that it prevents inappropriate gene expression and recombination events that could affect the genome stability as well as improve the lifespan of that particular chromosome there. So I hope I am clear. So anyone has any doubt there? Okay, so I think I am clear. So if you have any doubt, you can just type it out, or you can unmute yourself and ask, no issue in that case. Okay, so I will just move on to the next question. Coming on to the next question. Poulting chromosomes are found in which organism? Secretory cells of fly larvae, amphibian oocyte, in reptiles, or all of the above? Poulting chromosomes. Okay, I'm getting option number 2. Any other option? Anyone has any other option in mind? Okay, so answer to this question is option number 1, secretory cells of fly larvae. So amphibian oocyte is also associated with some chromosome, but in case of Poulting chromosomes, like these kind of chromosomes are associated with secretory cells of fly larvae. So if you come into option number 2, that those are called as Lamb-Rush chromosomes there, right? So both are similar in this case. What happens is that these are chromosomes that are much bigger in size as compared to that of a normal chromosome there. So you can see that this is a kind of a Poulting chromosome there, and also if you compare onto the size of other normal mitotic chromosomes at this scale, you can see the size is much more larger at this case. If you put a small scale bar here, you can see that it is much higher in size there, and even above the size of this 20 nano micrometer scale that is given here. So what happens is that these are found in secretory cells of fly larvae. So you know the process of DNA synthesis and cell division there, right? So DNA replication is followed by cell division. So in what happens here is that DNA replication happens, but cell division does not happen. So what happens is that as it undergoes multiple cycles of only DNA replication without cell division, they become larger in size. So this is the mitotic chromosome that is seen here. It undergoes DNA replication along with the DNA cell division. So it is much smaller in size because the size keeps in check. But here only DNA replication happens. So it keeps on replicating without cell division, and it becomes much larger in size. And what happens is that all the chromosomes that are present near close to each other start to aggregate to each other and forms this particular structure, what is called as a Poulting chromosome. You can see that there is some remnants of chromosome 3 there. X chromosome is also present there. Chromosome 4 is there, and other chromosome 2 you can also see there. So these are all present, the homologous chromosome of chromosome 2 and also chromosome 2 and chromosome 3 starts to interact with each other and aggregate to each other to form what is called as this polypeptide chromosome. You can also see the dark and light bands that are present there, right? And also you can see one chromosome there. So that actually integrates each of the chromosome arm there. So you can see that the chromosome is densely packed there with the DNA molecules and everything. And you can see that the regions where the two homologous chromosomes are separated, the chromosome homologous regions are also found close to each other in this case. So because of the repeated replication that is happening in this case without the proper cell division, these particular giant chromosomes are formed there. Similarly, Lamb-Rush chromosomes are there, which are found in case of amphibian oocyte. However, polypeptide chromosomes, these kind of structures are found in secretory cells of fly larvae there, and is much bigger as compared to that of normal mitotic chromosomes. So I hope I am clear. So anyone has any doubt in this case? Okay, so I hope I am clear. So I will just move on to the next question then. So coming on to the next question, pick the incorrect statement about SMC protein. They are conserved. They can fall back into themselves via antiparallel covalent interaction. Two SMC proteins interact with each other at the hinge region, and none of the above. Which is the incorrect statement about SMC proteins? Okay, I am getting option number 2. Okay, I will just tell you the answer. I suppose option number 2 is correct. The answer incorrect statement is none of the above. All the statements that are given here are correct. So I will just go into a previous slide. So first option is they are conserved. We can discuss it much better in this case. Okay. So this is one of the SMC protein. This is a coercin structure of the coercin molecule there we have discussed before. So if you look into one of the SMC protein, take SMC3 there, right? So what happens is that this actually these SMC proteins are highly conserved in nature. That's one of the things. It does not show much variation within the each and every organism. And if you look into a second option, that is it can fold on itself to form this dimeric coil-coil structure. So if you look into a dimeric coil-coil structure, it is basically right. You can see that this structure is actually coil-coil structure, and it is dimeric in nature. There are two chains there, right? And this chain arises from a single polypeptide molecule itself. So if you linearize this particular SMC protein, suppose only I am considering SMC3. If you linearize this particular protein, I should have shown one individual image here. So if you linearize this particular SMC3 protein, it exists as a large polypeptide there. So this particular region is called as hinge region there. It comes in between. So one of this coil actually extends into this region with the head region there. This is the head region there. So hinge region there. One polypeptide extends this region, and there is head region there. And the other polypeptide actually extends from this hinge region to this end, and the head region actually ends up this way. So the overall structure is like there is a linear molecule which consists of head region at both ends and the hinge region in between. And this polypeptide then can coil itself. This polypeptide from one end coil itself with the other polypeptide with the other end to form this dimeric coil-coil structure and the head region at this both end. So the second option is also right. First option is that it is conserved, it is highly conserved in nature, and second option is that it can coil on itself to form a dimeric coil-coil structure there, right? So that is also right. And also other option, I can just move on to So these two SMC protein interact with each other at the hinge region. So so I will just show you at this region. So this is the hinge region, right? So one SMC protein actually can interact with the hinge region of other SMC protein to form this hair clip like structure. So that option is also correct there. So if you look into the option, first option is right. Okay. So the first option is right, it is highly conserved. It can fold back onto itself by anti-parallel coil extraction, and two SMC protein can interact with each other in the hinge region. So all these three statements are correct in this case. So in this incorrect statement is none of the above. All of the above are correct in this case. So does anyone have any doubt? I hope I am clear in this case. Should I repeat it once more? I could have shown you a much better diagram here, however. Okay, no issue in this case. Okay, so I think it's clear. So I will just move on to the next question. Okay, TATs are full form of this. We have, okay, sorry, I should have associated this particular diagram here. So okay, so TATs are basically in this case topologically associated domain. From this you will know the answer. So for that I have to arrange this presentation and all. So does anyone have any doubt in this case? I will just explain it some of the description of TATs there. So topologically associated domains or TATs are fundamental units of three-dimensional nuclear organization in the genome. So these are specific regions that are present within this region there. Basically these are recognized with this coercive molecule or motor proteins there, and it is bound by this CTCF regions there, right? So CTCF proteins there. So it actually prevents the loop extrusion up to from a particular region there. They are self-interacting genomic regions there, where DNA sequences with the TAD interact more frequently with other sequences with outside that of TAD. You can see that there are several structure interacting regions that are present there. So they can basically interact with each other, and they play a crucial role in regulating gene expression by limiting the enhancer promoter interaction to within the domain. So in order for a gene to express itself, the enhancer and promoter should be in a close proximity with each other. Then only transcription factor can bind and then undergo a gene expression there, right? So TATs actually play a crucial role in regulating gene expression by limiting this particular interaction. So that's one of the other function of TATs there, and also they provide as a structural scaffold for regulatory landscape, facilitating interaction between these regulatory elements, that is this promoter as well as enhancer there and target promoter there. And also TATs are conserved across species, suggesting their importance in genome organization and as well as function there. And also these are conserved around different cell types and species. These are enriched with certain proteins like CTCF protein or TTC binding factor there, right? And other what is it? 1 minute. And other coercive molecule, because this we have discussed before. TC binding factor is actually a binding factor that binds to this TAD region and prevents the loop extrusion that function that is carried out by this coercive molecule there. So that's one other functionality of this TAD domain. Apart from that, this can interact with each other, and it also has the specific capacity to regulate the enhancer promoting interaction, thus affecting the gene regulation. And also it promotes a landscape for binding for this CTCF factors there, so that it can tell the coercive molecule that loop extrusion should be finished right now, and then it will be affected by this this Whopper proteins, and also the coercive can release out of that particular region. So yeah, so I hope I am clear. Sorry for this question. I just forgot to arrange this slide. So does anyone have any doubt in this case? Okay, so I think I am clear. So I will just move on to the next question. Okay, coming on to the next question. Coercion is thermoregulated by which protein? MMS21, RAD to CIR1 or MCD1. So mylation is also a type of modification there. So which type of protein does that to coercin out of these four options? Okay, so I think I will just give you the answer. So the answer to this question is 21. So basically it's a sumo E3 ligase that is a part of SMC5 or 6 motor protein complex. It is also a SMC complex there, right? Which is involved in DNA repair and recombination process and everything. And it plays a crucial role in somatic coercin. So it coercin also has a other functions like clearing of DNA damage and DNA repair process there, right? So this particular MMS21 is a sumo E3 ligase that actually promotes the summoylation of coercin molecule, and then it can undergo this DNA repair process and everything. So several modification that is present in coercin molecule actually carries out different possible functionality. That is it is multifunctional protein there, right? So basically this MMS21 actually acts as this particular E3 ligase which carries out the summoylation and all, which is involved in DNA damage and DNA repair process. So anyone has any doubt in this case? I hope I'm clear. Okay, so I think I'm clear. So there's no doubt. So coming on to the next question. What is a histone fold? Two alpha helices connected by two loops, three alpha helices connected by three loops, three alpha helices connected by two loops, none of the above, which may be the answer. What is a histone fold? It's a part of that histone octamer and everything. How that interaction three-dimensional structure comes to be. How many helices does it have? If that's so, how many loops does it have? If you look into each and every histone moiety, right? Histone 2H, 2H, 3H, 2B, each and everything has this particular predominant structure there. That's why it's called a histone fold. Okay, so the answer to this question is 3 alpha helices connected by 2 loops. So this is the basic structure there. You can see that there is H3, H4, H2A and H2B there, right? So there is a particular structure that is denoted in between, that is the histone fold there. And this broader regions that you see here there, that are represented as helix, alpha 1, alpha 2, alpha 3. So there are basically 3 helices that are present there. And in between you can see the loop region that is present there. That is, in each case there are 2 loops there, right? 1 or 2, 3 alpha helix and 3 loops. So even if you look into a PDB structure of that particular helix, so this is one histone octamer, histone molecule, this is the other one. So you can see that so this is the H3 one and this is the H4 one based on the color there, right? So you can see that this is one alpha helix, this is another alpha helix, and this is the third alpha helix. Similarly, in this case also H3, you can see the one alpha helix is there, one second alpha helix, third alpha helix, and each of the alpha helix is connected by a loop region there. There is one loop there and second loop there in case of H3, and in case of H4, one loop there and a second loop there that connects the alpha helices together. So if you look into a structure of what is called as a histone fold, it consists of a 3 alpha helix connected by 2 loops which are conserved in each of the three histone core histone molecules. There is H3, H4, H2A and H2B. You can see the diagram there. So that's the basic answer there. So anyone has any doubt in this case? Okay, so I think it's clear. So I will just move on to the next question. Coming on to the next question, which of the following modifications are found in histone? Acetylation, methylation, phosphorylation, citrullination, all of the above, or none of the above? Which type of modification are found in case of histones? Yeah, basically you are right. So the answer to this question is option number 5, all of the above. So yeah, so in case of acetylation, if you so it involves the addition of acetyl groups to a lysine residue that is present in histone. So in this case, it neutralizes the positive charge of histone that are present in histones there, right? So it reduces the interaction between the histones and DNA, leading to more chromatic structure that facilitate transcription. So in most of the cases, acetylation actually promotes a more chromatic open chromatic construction there, right? So in that case, it actually promotes gene expression. It increases the gene expression, that is promote transcription there. So it is catalyzed by histone acetyl transferases as well as histone deacetylase. In case of methylation, it can be either added to methyl group can either be added to lysine or arginine residues there. And it does not affect the charge of the histones there, but affect the transcription by recruiting specific proteins that recognize this modification. So it can either activate or repress transcription. In case of acetylation, it leads to majority of the time it leads to increased transcription. But in case methylation, it can either activate or repress transcription. So it doesn't alter the charge that is present on histone, but it helps in recruiting certain proteins that can either repress or activate transcription there. For example, this particular methylation that is happening at this H3 lysine 4 region there, you can see that it activates promoter, but this particular methylation that is present in this lysine ratio actually represses genes there. So in this case, acetyl transferase carries and deacetylase carries out the process. In this case of methyl transferase and demethylases carry out this process. So that is the difference between acetylation and methylation there. In case of citrullination, what happens is that it is carried out by this PADs or peptidyl arginine deaminases. It can alter the gene expression by converting arginine residue to citrulline. So arginine can be converted into citrulline by adding this particular citrullination there, and it can similar to methylation, it can either activate or repress transcription depending on the concept and specific residue that is involved. For example, this particular situation at this histone 3 modification actually activate gene expression by citrullination of other gene residues may repress it. And histone phosphorylation finally, in this final modification there, affect the chromatic structure by altering the electrostatic interaction between histones and DNA, because this phosphate group is a negatively charged group, right? So as in comparison to this acetylation and all, it also affects the charge of the DNA molecule there, thereby changing its chromatic compaction and accessibility. So for majority of the cases, phosphorylation actually reduces the histone DNA interaction, making it open to DNA transcription and everything. So however, certain phosphorylation events such as this particular modification during mitosis are associated with chromatin condensation. So it's not like if you are getting phosphorylated or acetylated, it can always increase the gene transcription there. Somehow at some points, at some amino acid modification can also repress it. But majority of these cases, it actually makes the chromosome more open and thereby, what you say, increasing the rate of transcription there. So that's the basic difference there. So I hope I am clear. So anyone has any doubt in both these cases, in all the modification that we have just discussed till now? Okay, so I hope it's clear. So I will just move on to the next question. Coming on to the next question. It's a basic one. Which technique helps you to identify specific DNA sequence bound to histone? FRET, FRAP, or co-IP, or fluorescence microscopy? Okay, so the answer to this question is co-IP or co-immunoprecipitation. So do check that out. What is the technique? What kind of technique it is, and it is actually given in the lecture video, so you can refer that and get to know about it. Yeah. So I will just move on to the next question. Okay, coming on to the next question. To count the number of SMC 2 or 4 that is condensing molecule complex at the loop neck using a double threaded DNA assay, which of the fluorophore was used? Atto 647N, GFP, RFP or YFP or none of the above. It is in the third lecture video, basically which type of fluorophore was used to count the number of condensing complex and the loop extrusion process. Yeah, I'm getting option number 1. Any other option? Yeah, basically you are right. It's actually a fluorophore basically in this case. So it gives a kind of a reddish to orange kind of color there. So based on that, the lifetime of this particular fluorophore is quite high as compared to other fluorophores that are present there, and also as compared to GFP, RFP or YFP, these are not that bulky group. These are all kind of bulky group. GFP, green fluorescent protein, red fluorescence or yellow fluorescent protein, these are all kind of bulky in nature. But Atto 647N is not that kind of bulky and hence can be act as a fluorophore, and it also has a high like quenching lifetime, because a fluorophore after a long period of time upon exposure to a laser start to quench out. So it has kind of a higher lifetime as compared to a other fluorophore molecules. So it was used to count the number of condensing molecules that are present within the loop region of the or the neck region of the loop extrusion DNA complex there. So I hope I am clear. So anyone has any doubt in this case? So okay, so if there is no doubt, we will move on to the next question. Okay, coming on to the next question. Which of the following statement is not true? Cystochromatic cohesion promotes proper bipolar attachment by steric constants and contention. Without condensation, lagging chromosomes can be cleaved by during cell division. The cohesin complex generates cystochromatic tension cohesion by tethering cystochromatic. All of the above or none of the above? Yeah, I'm getting option number 5. None of the above. Any other option? Okay, so the answer to this question is option number 5, none of the above. So all the other options are correct. Cystochromatic cohesion, like this particular steric constraint and tension that is created within the cystochromatid, actually promotes the bipolar attachment. Like it helps to promote the mitochondrial spindle attachment, and then because of the tension that is created there, the cystochromatids can get separate from each other. And also the based on that theory, it is also correct that option number 3 is also correct. And without condensation, lagging chromosomes can be cleared out during cell division. Lagging chromosomes, which are obviously part of Okazaki fragments there, right? So without that particular condensation, the DNA gets degraded out by the activity of several DNA denaturing enzyme or DNases there. So with condensation, that particular lagging chromosomes cannot be cleaned out there. So that's also a correct answer. So if you look into this particular option, none of the above is the probable option to this answer. So I hope I am clear. So anyone has any doubt? Okay, so I think I'm clear. I will just move on to the last question. Okay. So the coercive molecule is simulated by this we have discussed before, MCD1, NSE2, MMS21, SEC3 or NSE1 or none of the above, which may be the answer. You will get to know one more answer to this question. Basically, we have discussed this before, right? Okay, so the answer to this question is MMS21. This we have discussed before. Other than that, one other homologue is also there, NSE2. So, yeah. So, NSE21 is a sumo ligase associated with this particular complex, SMC5 or 6. And is crucial for simulating coercing complex including SMC1, SMC3, SMC in response to DNA damage. So, whenever DNA damage is there, this particular complex is getting this particular SMC proteins are getting simulated and then it can carry out DNA damage repair processes. And also it helps for maintaining the cystochromatic cohesion and also, as we have discussed, facilitate the DNA repair process and all. And yeah, it binds to the coiled arm of SMC5, position is for somalia activity, and because of the ATPase activity of SMC5 or 6 complex, the head region can actually undergo ATP hydrolysis there, right? It is needed for regulating the summoylation ligase activity of this particular MMS21 or NSA2 complex there. Binding of ATP to SMC5 facilitate conformational changes that enhances the summoylation processes. And also it specifically targets and summoylates subunits of the coercing complex such as RAD21, STAC2 using SUMO1. And it is, yeah, as we have discussed before, for cystochromatic cohesion it is involved, and also in case of DNA repair process and everything. So I hope it is clear. So anyone has any doubt? Okay, so I think there are no doubts. Yeah, this PPT will be okay. Excuse me. One. Yeah, all these sections that I have presented till now, right? So all of the PPTs and recording will be shared with you. The recording will be presented YouTube. You will get the link, and also the PPT will be shared in a Google Drive. So I have done till 5 week presentation. So all the 5 week 5 presentation I have uploaded in Google Drive. So this will I will upload it by today itself. So you will get the link and you can just check it out. So yeah, so yeah, you will get all the links basically for the PPT as well as the recording. Okay, so I think if there are no doubts, then I think we can leave the section for today. So thank you for joining. So we will meet everyone by next week.